And we are recording this, and it will get posted to our webpage when the seminar is over. Let's see. Since we're doing this seminar remote, this is the second of 9 Groundfish seminars running through December 14th. Since we are all doing this remotely, the speaker will use a pointer or be descriptive when indicating specific points on the slides because we can't see where the arms are pointing to. And to help with this format, please mute your audio and turn off your video feed to avoid additional distractions. Also, Liz, please keep your questions for the end of the seminar so the speaker isn't interrupted, or type them into the chat box and Liz and I will compile them. Before I introduce today's speaker, I want to remind people to join us for next week's speaker, Carolyn Sinet from the Department of Fisheries and Oceans in Canada, who will talk about filling science gaps in response to an unprecedented increase in redfish biomass in the Gulf of St. Lawrence. This will be held at the same time next Tuesday, October 19th. So for today's speaker, Jim Thorson leads the Habitat and Ecological Processes Research Program at the Alaska Fisheries Science Center, which involves envisioning future research and partnerships regarding essential fish habitat and loss of sea ice. He hopes to encourage further synthesis of direct and impacts of fishing on population status and productivity. He also collaborates with researchers in all AFSC divisions to integrate monitoring, process research, and modeling efforts to respond to ongoing changes in climate and research resulting habitat. Today, Jim will tell us about the grand challenge for habitat science, synthesis of fishing effects, stage structure dynamics, and movement.
Yeah, thank you, Mark and Liz. Clearly, I wrote that bio while I was multitasking. I'm not sure it syntactically made a lot of sense at points, but you get the idea. And I hopefully have met many of you since I started about 3 years ago at the Alaska Center. Before I forget, I've had the privilege of learning a great deal about the Alaska Fisheries Science Center and learning about our, diverse and well-integrated research regarding habitat. Part of that is supported through the Essential Fish Habitat request for proposals that HEPR administers with funding from the regional office and the Science Center every year. That RFP is due at the end of this month. It's about $350,000 a year, $350 or more. And so, I'm excited to talk with people now or later about proposal ideas. So I'll be noting co-authors in each of 3 sections. And, the intent of this is to kind of continue a conversation with the group about, how we can essentially, how we can connect habitat science with all the other science occurring at the center and how we can do it in a way that is impactful to management and therefore likely to kind of continue to get well funded. So I've been calling this the grand challenge for habitat science. And I've got a few kind of toy demos, not of sort of how to solve the grand challenge, but of kind of approaches that colleagues and I have kind of taken to kind of pick at the sides of it. So thank you, everyone. Let's see if I can, So now I can put an arrow, but I can't go forward. Does anybody, I should have tested the page forward thing before.
In the lower left-hand corner. Oh, there we go.
That's fine. As long as I get rid of the annotation, it looks like I can do it. There's no need for me to annotate anything, so I'll just ignore that. As I said, I'll start out by introducing what I've been calling the Grand Habitat Challenge. And then these kind of 2 little toy demos, one about individual fine-scale movement and one about how anybody at the center could pick up our extensive stomach content data and use it to complement what they're doing for habitat. So first up is this paper that was published last year with co-authors Al Herman at PMEL, Kevin Sawicki at ABL, and Mark Zimmerman here at GAP. the grand challenge for habitat science, stage-structured responses, non-local drivers, and mechanistic associations among habitat variables affecting fishery productivity. So I guess my cartoon is that we, over decades, we at the Alaska Center and also many other science centers have been doing these sort of pressure indicator response approaches to habitat studies. So we'll typically do a first set of studies that look at sort of impacts of fishing on, on habitat features. It could be, benthic infauna, it could be structure forming invertebrates, corals and sponges. It could be mangroves, impacted by development. Each of those, typically will do, either an observational study about impacts, or we'll do, some kind of experimental backy before after control impact design for a given type 1 and type 2 error rate to detect a significant impact on a feature of habitat. And then if we find a significant impact, we then try to associate that feature that's impacted with resulting density of juveniles for mangroves, or condition of fish for structure forming invertebrates, or a hodgepodge of things for benthic infauna. And so this sort of two-stage approach, I mean, it's first of all, it's subject to all sorts of kind of user error. In the first study, it takes experts to, it takes a lot of brilliance to set up a design to detect impacts on these habitat variables. And then a second study is required that is often also very difficult to design. So for instance, I'm not aware in the Alaska region of any empirical study having definitively demonstrated fishing impacts on structure forming invertebrates, infauna, or, nearshore habitat. Instead we assume something a priori about that, which is a reasonable assumption. And then we are still putting a lot of work into the second type of study about how these structure forming invertebrates will affect condition. Supports a lot of interesting science, but we haven't finished the stack for any single pressure indicator response. And so our capacity to inform management is limited. And of course, the problem is that, things are way more complicated. So, fishing is not just, impacting habitat variables. It's also, it's impacting predators, shown here with this cute little orca. Humans are simultaneously doing this unreplicated experiment of ocean warming. And all of these are kind of matriculating through as direct and indirect impacts. And we are responsible, ethically and also in statute to assess all of these in essential fish habitat. And so just to think this through, fishing could result in, many things. Some of them are listed here, kind of categorized, and, here's how you could study it. So fishing could result in depleted benthic communities. That could result in less benthic forage and structure and affect change, fish productivity. And that's essentially the kind of main thrust that we've been doing in the SDM species distribution models and the fishing effects model at the Alaska Center. Fishing could also result in a depleted midwater surface community, resulting in less pelagic forage and changing fish productivity. It could result in less nursery habitat, resulting in less recruitment. Those are all indirect or habitat mediated effects, but of course, my claim is that, we also could be studying direct kind of exploitative effects. So, instead of doing this two-stage study of looking at fishing on infauna or structure forming invertebrates or nearshore habitat, we could look at direct localized depletion of fish in good or poor habitats. And how that changes the distribution among good and poor quality habitat, and how that affects resulting, per capita growth rates or productivity rates for the populations that we manage. Fishing also directly affects the composition of target and bycatch species, and that results in changing potential for attainment given management limits. And so, a lot of times, when people talk about habitat effects, in our management system, it's about closures. It's about like the red king crab closure area in Bristol Bay. And for instance, that closure is affecting the composition of target bycatch species. It's affecting the attainment given our management limits. And so that's a direct or exploitative effect of fishing on productivity for these different species. That's not the type of habitat research that everybody wants to do, but that is a type of habitat research. And it's one that could be done much more directly than the kind of two-stage study that we have been doing. So, to think this through, I guess the Grant Habitat Challenge is specifically to try to formalize a framework for thinking about how localized management decisions, decisions about fishing affect population-wide productivity. And to think that through, there's sort of two schools that we use here at the Alaska Center. One is sort of how we do stock assessments. And so in this case, we have a state-based production model for the North Sea. This is a paper in Fish and Fisheries that's continuing to kind of affect things in Europe. And at the bottom here, we have these sort of fits to data for two stocks. And we can define stock status for these two with respect to different targets. And the tradition of doing stock assessment is to have a model. We have data, we have process research, and it connects through a model to management, in which we are trying to understand changes in biomass over time as affected by density dependence, environmental responses, and sort of the direct offset of exogenous fishing. And so we have the effect of current biomass, environmental predictors X, and sort of a human impact C affecting changes in biomass. By contrast, in habitat, we often propagate our monitoring and process research through these species distribution models, or SDMs, and they're used widely in ecology. They include fine scale variation covariates, and they're usually, the response is biomass, not a change in biomass, and it just predicts that based on environmental responses. So it doesn't directly represent harvest or impacts, and it doesn't directly represent density dependence. So, research in the last 5-10 years has generalized both of these, have kind of combined them to have a process that looks at changes in biomass at each location S, S1, S2, S3, or 3 different locations or grid cells in a raster. And the change in biomass is predicted by local density, by local catch, and environmental variables with an estimated environmental response. And so, this has all the variables of both a stock assessment model and a species distribution model, and it could, be used to define, targets and stock status at each location within a stock's range. So I've been calling this the spatial biomass dynamics model, and it's sort of a conceptual model to support this grand habitat challenge. Of course, this sort of cartoon doesn't do a lot of things that we might actually want it to do. I'm sorry, this is a slightly old slide. I kind of reduced this down to 3. One of them is stage structured effects. We know that fish have demographic rates that vary with size, and that the size structure provides useful information. We have non-local effects like individual movement and ecological or environmental teleconnections that are not accounted for in that kind of simple spatial biomass dynamics kind of conceptual model. And then there's a bunch of questions about how do you attribute changes to direct and indirect pathways, and how do you predict into counterfactuals like novel climate conditions. And so each of these is a challenge to that sort of simple cartoon model that I'll talk about how a person could deal with. So the first is this sort of idea of stage structured responses. And just to give a concrete example, here's different variables that we've produced for Pacific Ocean perch at the Alaska Fisheries Science Center. At the top is juvenile occupancy and density. The bottom is adult density, and then the right-hand side is adult growth potential. So, each of these has a different sort of monitoring design, a different analytic design, and then results in a prediction. But of course, if we want to talk about POP and how, for instance, like, a new shipping route or a new fishing operation or a proposed, oil pipeline could affect POP or any other species, we don't want to think about how it affects juveniles separate from adults. We want to think about how it affects POP as a whole. And so we have to kind of integrate across these different efforts. And so, oh, shoot. I thought I updated this slightly. Okay, that's fine. An alternative approach is to, basically define these rates at each location. And so, as I showed, we could have, information about survival across space, or maybe we assume that it's constant across space is kind of a null hypothesis. We often measure length at age across space and weight at length. We have direct measurements of. We could have some information about reproductive output, maybe not for POP, but for some species we have information about, egg production as a function of individual size. We have information about skip spawning for some species. And so in some cases we could fill in information about this, and other ones we can't. And then finally, like reproductive survival and advection, sometimes we have a modeled product like an individual based model. People don't always agree about the individual based models we have, but it is a basis for trying to link up, production, in the Southeast and how that might affect the, recruitment in the central Gulf of Alaska. That's a bad example, but just a hypothetical. So once we have all these different sort of rates defined at every location across in this case, the Gulf of Alaska, we could either combine them by calculating an intrinsic growth rate of, little r, at every location by making a Leslie matrix at each raster cell, and little r, is dimensionless, and it's sort of a common currency that's often used in ecological theory to think about, sustainability and persistence at a community level. And so that, that could be a common metric for defining EFH that integrates across life stages and demographic rates. Alternatively, in some species, maybe we can parameterize an individual-based model with all of this, a full life cycle individual-based model that you run to equilibrium, and then you can define a production at every location as the change in total abundance that would occur if you eliminated that. So you could do kind of a sensitivity analysis on that IBM to get at how important every location is. In terms of non-local effects, I'll talk first about this, and then we'll talk about movement in a minute. I and others have been thinking a lot about kind of ecological teleconnections, so essentially how to use observational data to look at how something at one location affects density or productivity at other locations. One way to do these sort of, estimate these teleconnections is to, that was developed originally by physical oceanographers, and atmospheric scientists, is to fit these sort of empirical orthogonal functions. So in this case, if you fit an EOF to bottom trawl data in the Eastern Bering Sea, you get sort of a mode of variability that pops out of the data. That mode of variability is highly correlated with cold pool extent, even though that cold pool wasn't explicitly in the model. It pops out because each of these species has sort of an idiosyncratic response to, wintertime sea ice production and resulting cold pool extent. Whether it's a direct effect or whether it's because of how cold pool affects the timing of copepod diapause, or, the strength of the, Bering Sea greenbelt. It doesn't exactly matter. The point is that there's this kind of synchronous ecological teleconnection that's simultaneously affecting productivity across a whole landscape. And so in this study, we kind of walk through how each of these species has a response to cold pool, and show sort of how to estimate these sort of teleconnections. And then finally, the third one is about sort of how do you deal with multi-causality? Like, how do you deal with the fact that there's a bunch of things that are changing all at once? And so, a person might wonder, are copepods more important than other forage for winter young of year, age 0 pollux growth. And is RNA DNA hypothetically a better proxy for for foraging success than morphometric condition? And so in this case, you end up with a bunch of these different sort of variables that you can measure. Here I'm showing kind of 9 related to an age 0 pollock foraging module. And essentially you're trying to estimate the strength of different arrows shown on this, like how much do copepods versus krill affect a direct measurement of forage success in terms of stomach energy densities, and then how much are stomach energy densities, which is sort of expensive to measure, how much can that be indirectly measured using RNA DNA measurements or morphometric condition or bomb calorimetry or whatever else it is. So in this case, there's something called a structural equation model, and basically it's sort of a matrix of effects. So you form this matrix in the bottom right, where some variables are thought to have no effect on other variables. For instance, you might have water clarity is assumed to not have any causal effect on surface temperatures. They might both be driven by the same thing, but ignoring that for now. You might think that water clarity has no direct effect on stomach energy densities except via krill or copepod or other prey densities. And so anytime you know that there's no direct causal effect, you put in a 0, and then you basically come up with a list of hypotheses, hypothesized direct interactions or causal effects, and try to estimate those coefficients. And you can do that using what's called a structural equation model. I've got some of the math up above. We don't really need to go into it, but it's basically a way of parameterizing a covariance matrix and then fitting it as a likelihood model. So, and I can share the script for showing how to do this, how to generalize structural equation models in this way if anybody's interested. So, and then finally, these kind of conceptual questions we have, like, are copepods more important than other forage for winter young of year growth? You could end up asking questions about, like, oh, is the direct effect of copepods on stomach energy density, is that coefficient greater than the coefficient for krill on age 0 stomach age 0 prey densities in a stomach? And is RNA DNA a better proxy than morphometric condition? Again, you could express that as sort of a comparison of coefficients in this model. So, that's kind of a quick description of this sort of challenge that I want to set up of how do we want to predict the impact of localized human activities on population scale productivity. My suggested approach is to combine concepts from stock assessment and habitat analysis to directly include proxies for human impacts, caches, habitat disturbance, condition your predictions on current and future environmental conditions, and include both of those to predict changes in habitat specific productivity. So those are the kind of things that are included in that sort of spatial biomass dynamic model. And of course, doing it that way results in many, very obvious challenges. So stage structured responses we can resolve by assembling stage specific habitat responses and synthesizing across them using a common currency. Non-local responses, we can either explicitly model movement or identify modes of variability resulting from ecological teleconnections. And, mechanisms, these kind of multi-causal or these kind of multiple things that are changing at once, mechanisms that link environmental variables, we can resolve using structural equation models, or using essentially like a regional ocean model, ROMS, as a structural model for the relationship between environmental variables. So, hopefully that shakes loose some ideas for people. Next, I'm going to move to the kind of two toy examples, that again don't sort of solve this grand habitat challenge, which would take effort, concerted effort from people across the center. But instead, these are just kind of demos of sort of how some of the concepts could be tackled. So in this case, this is a paper that I did with, Steve, Dan, Kelly Kearney, Ned, Julie, Matt Siski, who's a postdoc at UW, Kevin again, and Grant Thompson. It's published in Fish and Fisheries this year, looking at high resolution movement rates estimated from environmental variables, tags, fishery CPUE, and surveys. And so, yeah, I tried to strip out a lot of the math here. So I guess I want to start by pointing out that there's a lot of different data that's informative about movement. There's conventional tags. We at the Alaska Center have an amazing conventional tag data set, kind of like Floyd tags. They have low return rates, and their return is dependent on fishing effort, but we can resolve those issues. We also have an amazing database of surveys. We have fishery CPUE, which requires some additional assumptions to interpret, but is season-wide. We increasingly have a lot of archival tags, and with the increase in sablefish, maybe we'll have a future in which we have more money coming out of our sablefish program for archival tagging. We increasingly have movement gates, either weirs in freshwater systems or upward-facing acoustics, and those can basically measure the flux of individuals across a fixed location. We have selection studies, so like Ben Laurel did a lot of work that I admire a lot about, how different conditions will drive, habitat preferences, measuring those in the lab. We have chemical markers, stable isotopes, compared with an isoscape. We have genetic markers, parasite markers, and then finally, stomach contents data. So we have this tremendous database of different ways of inferring movement, and I would say, rarely manage to spend the time to synthesize all of those in a formal way to kind of inform our management process in cases, Cadillac cases, high priority cases, when we might want to do that synthesis. Perhaps part of the reason is that conceptually you kind of need a framework to do all this, and so that's what we propose in this paper. We decompose an instantaneous movement rate M star. It's a matrix representing a movement rate from every location to every other location. And we decompose that into a diffusion process, like random movement, away from, kind of searching or unmodeled kind of individual movement away from where they are. A taxis rate. So taxis is defined as sort of the movement towards preferred habitat. And then a passive advection rate. So obviously, plankton by definition are going to be advected passively along the kind of, gradient field, along the direction of, dominant currents and tidal currents. And so we could include, but of course they can also modify that passive advection by vertical, choosing vertical movement. So in part, it's sort of a known advection rate, and in other cases, they can sort of modify the speed at which they move along that field. Yeah, and so once you have this instantaneous movement matrix M star, you can integrate that across a known time interval using what's called a matrix exponential. And so just talking about this a little bit, instantaneously, a fish is never going to move from one location and skip. It always has to move through intervening locations, so within an instantaneous movement rate, if you have a raster, if you have a grid, a fish can only move to an adjacent grid cell. When you integrate that instantaneous movement process across a time interval, then fish there's a non-zero probability of them moving to other locations, any other locations. They don't need to be adjacent anymore. And so once you have that integrated movement fraction matrix M, you can use that to project your estimates of abundance from one time to the next time. And so in terms of math, if the columns of that movement rate matrix sum to 0 and all of the off diagonals are greater than 0, then that integrated movement matrix conserves abundance. It sums, all of the columns sum to 1, and none of them are negative, like there's no negative fish in the world. So it's pretty straightforward math. It's the kind of thing that anybody who's used to doing R could learn how to do with, a couple 5-8 lines of code. The key here is that taxus process can be modeled from a habitat preference function. So let's say you've got a set of habitat variables X, so like Ned Lehman right now, hosts these rasters of, phi and bathymetry, and we, of course, other people have developed all these products, Mark and Bob McConaughey and many others. We then make sure they're all in the same resolution that we want to use as a community, and we reserve them on that kind of fixed projection. And so you could get a bunch of those variables, and you could, try to figure out, is there a given sediment size phi that your fish prefers? Is there a given bathymetry that they prefer, or temperature, or whatever it is, light level? And so in this case, their preference is just the product of these environmental layers that we have measured, and then a bunch of unknown habitat preference coefficients, which I'm calling alpha. So you can make this habitat preference function for every location, every grid cell G. And then taxus is just the difference between the habitat preference at one location or another. That's the taxus rate. And that just goes into that instantaneous movement rate. And so taking kind of like a one-dimensional example, here's sort of a little illustration of just a one-dimensional habitat along the x-axis. And in this case, we've got depth, where it's getting deeper as you go from 0 to 6. And then there's a bottom temperature where it's coldest in the middle, and then just kind of making up some coefficients for this habitat preference in the top left. The resulting preference is shown in black. So it's half of the temperature plus the depth, but it just kind of prefers being in the left because it's kind of, near 0 because it's shallow, and it kind of, or because it's, because it's low temperature or high temperature. Like towards 0, it's higher temperature. It doesn't prefer being in the middle because it's low temperature, and then on the right towards 6, it's deep and it's high temperature in this sort of made up example. And so it prefers being towards 6. So using that habitat preference, we can define a diffusion matrix that's shown in B here, a taxus matrix where it's moving from blue to red. And then we can kind of solve at the bottom row, if they start at the gray bar in time 0, you can solve where they would end up after 1 time interval, or 3 or 10, or an infinite number of time intervals. As t goes to infinity, regardless of where they start out, they converge on this black dashed line, which is the kind of stationary habitat utilization function. That's sort of their resulting distribution at a population scale. And we can look at sort of how quickly they converge from their starting point on that distribution by comparing the red, the purple, and the blue across those different bottom panels. And so, the point, there's a couple things to take from this. One is that, we can connect habitat, we can connect environmental layers like temperature and depth to a resulting stationary distribution. That black line in the top left in A looks a little bit, but not identical to the resulting utilization distribution shown as the black line in the bottom row. They look similar, but not identical. Another point is that if we have an instantaneous movement process that we know or we estimate, we can then back out what's the utilization distribution, where on average would a fish of this population be, and we could use that utilization distribution, the black line in the bottom row, to define essential fish habitat if we, or inform essential fish habitat. So as an example, co-authors and I applied this to Bering Sea Pacific cod in the eastern and northern Bering Sea from 1982 through 2019, and looking at their distribution in 2 seasons, summer and winter. Summer, winter, I forget how I set this up. It's like summer, and then it'd be winter of the next year, and then summer the next year. It's kind of an accounting thing, but and then the covariates we had, it was bathymetry and summer and winter bottom temperature for ROMs. We had a summer bottom trawl survey and summer and winter longline fishery CPUE. We had conventional tags, and then we had longline fishing effort as an offset for explaining tag recapture probabilities for those conventional tags. So, we made a little toy R package called ATM, or Advection Taxis model, that fits this model. And in this case, I put in like a spline effect of bottom temperatures or bathymetry on habitat preference, and fitted that spline response, and then visualized it using these kind of standard R packages. During fitting, without doing it in a really formal way, I assumed that the bottom temperature response was the same between seasons, but the bathymetry response was different between seasons. Kind of, in some species that can make sense if they have like a summer versus winter, summer feeding, winter spawning bathymetry response. Although I don't claim that that's a great description of Pacific cod. So, fitting this model and then looking at the estimates of what would happen if you release a tagged fish kind of near the pribs in different time, in either in the left column, and releasing it at the beginning of the summer of 2007 so that it goes through a cold stanza, or releasing it at the same place at the beginning of summer 2014, so it kind of goes through a warm stanza, and tracking it from the summer to the next winter, the next summer, and then a couple summers later. The model is not picking up a huge difference between summer, warm and cold stanzas, as shown by their resulting predicted distribution over time. But there is some difference in their predicted movement between warm and cold. The model also emphasizes that the population's not panmictic. It takes a while for a cod to this sort of simulated cod to diffuse. I should say that these results are not including information from our really informative electronic tagging studies. We could bring those in the future, but to avoid kind of stepping on toes, we chose to leave those data out. And so in the future, this could be updated again with really informative data. In this case, we can look at differences in habitat preference in summer and winter, and the model is predicting, again, not claiming this is a great description of cod specifically, but it does show what the model can do. It predicts sort of a broad distribution in winter for many of these years. And then the predicted densities, in this case, log numerical densities in different years as well. And showing, like, for instance, by 2017-18, a movement of densities up into the northern Bering Sea. We've got these sort of metrics of model fit that seem to do fine. I won't spend a lot of time on this. And then, once we have this high resolution movement process and this utilization distribution, we can do these kind of scenarios to think about how much, we can coarsen the resolution to look at the movement fraction from the Eastern Bering Sea to the Northern Bering Sea, and from the Northern Bering Sea back to the Eastern Bering Sea. In general, if we coarsen this and look at kind of a 2-box movement fraction, we see that there's, throughout this whole time, a predicted fairly low rate of movement from the EBS to NBS, and a fairly high rate of movement back from the NBS if a cod got up there. But that movement from the NBS back to the EBS has dropped a lot over time. And that's what the model says is responsible for this kind of increase in the proportion of the population in the Northern Bering Sea. We can do these sort of sensitivity experiments of excluding data. So if we drop out all the fishery data and look at movement from that same location in the PRIBs, we get a picture of their utilization distribution or their movement after, I think, several years. I forget exactly. And it looks pretty similar to all the data. The proportion of EBS looks similar. If we drop the survey data, by contrast, the model really doesn't make any sense. It's kind of wildly wrong about thinking that there was a lot of fish in the Northern Bering Sea originally. And then finally, if we drop the tagging data, the rate of movement is estimated way too low. So even several years later, there's not much diffusion away from the PRIBs. And as a result, the estimates of movement we can get from the movement between the EBS and NBS says that very few fish would have moved in the Northern Bering Sea in recent years, which again is sort of not consistent with what we think has happened. So in this case, the survey and the tagging data are most important. The fishery CPUE is not doing that much. Then finally, we can take these coarsened movement fractions and plop them into this 2-box stock census model that Grant had proposed several years ago. And without movement, the proportion of the Eastern Bering Sea is shown in this gray line, and then the dots are these sort of best available science, survey data estimates of the proportion of the NBS. And so the gray line sort of is consistent with recent years where we think that there were a lot in the NBS, but of course we don't think that was the case back in the 80s and 90s. By contrast, if we take the movement fractions from this model and plug it into the assessment, it does a better job of thinking that the assessment model does a better job of agreeing that there weren't many fish in the NBS in the past, but the movement rate constrains them not to move into the NBS that quickly either. So, neither the blue nor the gray line is really very satisfactory for stock assessment purposes, and I think the plan team and SSC have consistently gone ahead with kind of a 1-box assessment model until we can get better information about, better synthesis of movement, perhaps by including electronic tags in a synthesis framework like this. So conclusions, for people that maybe got lost in the math a little bit, we have a ton of data at the Alaska Center. Different species have conventional archival tags, survey and fishery CPUE, acoustic tags with autonomous detectors like sail drones for red king crab. We have genetic markers. We have movement gates like acoustic telemetry or fine scale arrays. We have a lot of tracers. And then we've got kind of predators as samplers. John Pyatt talking about seabirds as samplers in the Aleutian Islands and so forth. So all of these are informative about movement. And if you care about movement in your species, I think that there's going to be some combination of data that is informative, that it is, for affecting management, it's worth trying to think about how to synthesize. So then finally, the third case study, or the second case study, the final piece of this talk, is work that revisions are back in review ecology, working with Yumi at USGS, Tal Levy at OSU, and Gretchen Roffler at ADF&G, and it's showing how to my belief is that anybody at the center should be able to use our decades of stomach content data both from the bottom trawl and from, surface trawls that EMA have collected, ecofoci and EMA, to get insight about, the foraging ecology of their species. And they can do that using generalized linear models. So if you can run a GLIM in R, you should be able to do stomach content analysis. So, food habits data, again, you've got stomach contents, either from necropsies, onboard capture and sorting, or wrecks like wash-ups of dead marine mammals. We've got behavioral observations like bill loads for seabirds, and we've got kind of biogeochemistry, stable isotope signatures and fatty acid markers. And each of these has, some combination of, visual, genetic, stomach temperature loggers, electronic loggers, etc. So we've got this sort of explosion of food habits data, similar to how a lot of other parts of our research are working now with electronics. And so there's, similarly a kind of need to expand how we do analysis and synthesis of stomach content data. So in the past, as I understand it, people a lot of times just did this sort of what, to me, I will say, seems like a silly approach of taking the % frequency of occurrence, just the average of data, and treating that as the, proportions of a diet. And so, of course, this has several problems. One of them is that spatially unbalanced sampling will cause bias. So, if you're doing necropsies of washed up marine mammals, those washed up mammals might be preferentially skewed towards ones that were close to shore and didn't sink. If you, a lot of people have unbalanced designs. If you have seabirds and you're looking at their bill loads, that is only going to represent their foraging near that area, and of course what they bring back to their chicks, etc., etc. So we need to deal with this sort of unbalanced sampling that causes bias. Alternatively, people do this hierarchical expansion of post-stratified samples. So, I think that's how REAM has been doing, the program REAM has been doing this for a long time with our stomach content. We expand stomach samples in these sort of post-stratified boxes, and then we combine those across space using the proportions of density in those boxes. The problem with that, is that it ignores fine scale heterogeneity. There might be clear differences between warm and cold water, and that's not built into this post-stratification. It also can't use continuous predictors very easily, like size or age. So a lot of times it's post-stratified by space and by size. But of course that ignores heterogeneity within the size bins. So, people have tried to develop these ways of doing models, like a Dirichlet model. This is kind of what, Cam Ainsworth and folk have been doing. You can put in a mix of stratified and continuous predictors, but it requires modifying your data kind of a priori to fit zeros and ones, and it's hard to interpret. And then what I'm proposing, and what I've got a little R package that I can share with people, is to do this sort of Tweedy generalized additive model where you can put in covariates. You don't need to modify the data, and you can just run it as a GLIM in R. And so the theory for doing this is that you've got, foraging is essentially a point process. You've got some unknown location for every prey, and that's a random variable. If you assume that individual prey are independent random, the counts at a given location, a foraging location, will follow like a Poisson process, a Poisson distribution. And then let's say every prey has a size mark. If that follows a gamma distribution, then you end up with what's called a Tweedy. A Tweedy distribution is the total biomass of all of the prey that would have been encountered in a given foraging trip. And then let's say you've got some thinning process, like you've got some, predators encounter a prey but decide not to attack it, or they fail in their attack rate, or they have some handling time. Those are all types of thinning in a point process. And so we can define foraging as a thinned and double marked point process. So conceptually, let's imagine we've got 3 different prey distributions where the location of prey, they each have their own distribution. shown in the top row. And so every prey is a point, and their size is the size of a point. And then we've got three different kind of central place foragers shown in red, green, and purple. So looking at those sites, the distribution of prey that they'd capture on a foraging trip around that location is shown in the second row. And then if we approximate those distributions as a Tweedy distribution, we get this sort of theoretical distribution shown in the third row. And then if we further approximate it using a Tweedy that we can fit really easily as a glim, that's the fourth row. And so basically the point being, without having the time to go through it in a whole lot of detail, that we can approximate this sort of individual-based foraging process as a Tweedy generalized linear model. So why do we care about all this math? I mean, the point is that I think that, you know, everybody doing habitat research, I'm assuming, will have some experience with ANOVA or, you know, basic kind of statistical kind of hypothesis testing methods. And I think increasingly there's sort of some convergence around using R for that because we can share it between people easily. And so if you can kind of take the next step of getting to fitting a glim in R, you know, I could share a package with you to fit stomach contents doing this. So the R package looks, or the R code needed for this looks like this. This is loading package and a package called MV Tweedy that is the Middleton Island tufted puffin stomach contents. I then, in this case, fit a GAM, which is in the second kind of row. In this case, showing, let's see, this is an oversimplified example. It would be just showing a constant proportion for every group, prey group. I can predict the response in this data frame out, and then I can visualize that using ggplot. And so if I do this with a smooth spline on year and visualize it, you can get these sort of temperature responses and prey proportions for every prey species that is in this database. And what it shows is that at Middleton Island, the tufted puffins had this sort of early oscillation between Pacific sand lance and prowfish back in the 80s and 90s. And then in the 2000s, more recently, there's been this sort of oscillation between Pacific herring and capelin, where herring has a higher proportion when temperatures are warm, and that's significantly positive. And both prowfish and walleye pollock have lower proportions, significantly lower proportions when temperatures are cold. So in this case, we've got sort of a very simple glim, kind of looking at prey proportions for tufted puffins. We did this for wolves in Southeast Alaska. I'm getting to the end here, and I'm unfortunately running out of time. But here's sort of a landscape of prey utilization. And I just want to point this out. I should update this figure. I'm sorry to any co-authors on it. Some of these islands don't have wolves on it, but you know, there are islands where their proportion in their diets is very high of marine mammals, other areas with beavers, other areas with fish. You know, so their diet is, you know, highly context specific. So the point is you can fit food habits as a GAM or GLIM using kind of standard R packages. You can put in covariates affecting prey densities, and it's easy, you know, it's just as easy to do as a standard regression in R. So, you know, before getting to acknowledgements, you know, my basic message is that we can develop these density maps for species across life stages, including movement. I think that we should explore as a community direct impacts of human activities on habitat-specific productivity. So not just looking at impacts on habitat and then subsequent impacts of habitat on species, but looking at direct exploitative impacts. And I would be very interested to talk with people about indirect impacts on forage using stomach contents. You know, we have, again, this whole buildup looking at infauna in the fishing effects model, and we apply it to species that seem to have no association with infauna. You know, that's the kind of thing that we could really quickly knock down by looking at stomach contents to kind of test our assumptions about whether infauna will ever be a useful predictor of human impacts for a given species. So putting up a list of co-authors here, and thanks again to Liz and Mark for organizing.
Okay, well, thank you, Jim. And I meant to add that we tried to work you in last year and it just didn't work out. The logistics weren't there. It's nice to finally get to your presentation this year. So thanks for your patience for that. I don't see any questions in the chat. Is there anyone out there who wants to unmute their microphone and ask a question directly of Jim? While we're waiting for folks, I wondered, you know, it seemed like a lot of the data sets you're accessing are all sort of partial data sets in terms of space and time. And I wondered, it's a lot of adversity to overcome. I wondered if you had any comments on that.
Yeah, I mean, I want to say again that like all of our science is a relay, and so you know, any of us in our lifetime are going to get kind of small data sets and do kind of small synthesis, and you know, we just need to trust one another to give each other credit and to kind of get it to the audience. You know, none of us will succeed without sort of the willingness to kind of, you know, not just do our own analyses, but put them out there so other people can do them. You know, I think we do that well at the Alaska Center. You know, as one example, you know, we've got all this sort of electronic tagging data that will be incredibly informative about movement in the coming years. You know, I'm really appreciative of the groups that are doing that and how they 100% deserve kind of the first crack at sort of what those will mean. And then I'm excited about sort of, you know, opening up access to the whole community to think about how to use those. You know, once you kind of get that whole handshake, you know, that whole relay done the first time, it's much easier to show the value of it and then get funding for it again. And I might have seen a question pop up.
I have a mundane question. Is it possible to get a copy, for us to have copies of your presentation? Because there's a lot of information on it. It's very information dense. It would be nice to be able to look at it again.
Yeah, well, thanks for attending. And I think I think there's a recording. And of course, anybody can email me with my email at the top there too to get a copy of the presentation if they want that instead of the recording.
Thank you.
Anybody else?
Well, maybe I'll just also again just remind people that the Essential Fish Habitat RFP is coming up, and there's still plenty of time to come up with new ideas and aim to submit them. And feel free to reach out to me if you want feedback before doing so.
Thank you, Jim.
Oh, sorry. This is Megzy. I have a question that I typed in the Q&A, but I can just ask it out loud too.
Yeah, go for it, Megzy.
Actually, it's two questions. One is, can you send me a link to the MV Tweety package? And the second question is, I really like all of these like trophic interactions. It makes a lot of sense to me ecologically to include predation interactions into the characterization of predator essential fish habitat. And I'm wondering if there's a way, like, how do you see those kind of short timescale interactions fitting into our like management structure for EFH, which in my understanding is like one snapshot every 5 years and these static maps that we provide in the reports.
Yeah. I mean, I think that there's sort of two, basic uses for that sort of trophic information. One is using predators as samplers, and I don't think we do that as much. John Pyatt's talked about that. We've had some success kind of showing that groundfish are a good sampler of herring in the Northeast Center. And then the other one that we do more at the Alaska Center is using stomach content as a measure of trophic flow, energy flow, carbon flow in multi-species models. In terms of the first one, there are some species where maybe we have better information from predators as foragers than we have direct sampling information about their distribution. So that's sort of the focus on the prey distribution. In other cases, like, trying to characterize condition, understand the, suitability of habitat based on their sort of the energy density, their foraging success at a given location. And then again, like trying to look at sort of habitat, whether a place is a good or bad for a given focal species, is going to be driven by how much predation there is. So, like northern fur seal recolonized and ate a lot of stuff around one of the pribs. We probably have good stomach content data for that, and that could be, that story could be played out in terms of essential fish habitat for whatever they're eating, and I don't know the details enough to guess at whether it's important for crab and the pribs or what.
Sweet, thanks.
Thanks for the question, Miggsy. And we've run over our hour, so I think we'll end the general meeting there. But if Jim, if you're available for a few more minutes in case there are any other questions, maybe we can hang out for just a little bit. Thank you, everyone, for attending.
Yeah. Yeah, and sorry, the other question was about MV Tweety, and the paper's still in review. It'll obviously go live when the paper gets accepted, and I'll send that. I'll just invite you to the private repo, Megzy, when we get off here for now.
Awesome, that is great. I did a brief and frantic Google search for it while you were talking. And this makes sense. Great, thanks.
Yeah. Well, thanks everyone.
Well, you're getting a lot of thank yous in the comments, and so thank you so much, Jim. Great presentation, and hopefully people can catch up with you directly if they want to chat more about all this.
Sounds
Thank
good.
you.
Thanks again. Yeah, bye.